created by MindStick's Foundation Trust. Welcome, students, to today's Explainer. We are going to go on a truly fascinating biological journey together. We'll start with how we fuel the human body, look at the critical role of essential vitamins and water, and then dive deep into the basics of cell division, covering both mitosis for growth and meiosis for creating new life. Let's get started, shall we?
First things first, fueling the human body, from the macro down to the micro.
Think about the food on your plate. When digested, the proteins from our meals are broken down into amino acids. These are quite literally our body's fundamental building blocks. They are absorbed and transported through the bloodstream to every single organ and cell. And what happens next? The body uses them to create a vast array of necessary proteins. We're talking about melanin that gives your skin its color, the flexible myosin in your muscles, and even absolutely crucial hormones like insulin in the pancreas.
Now let's compare that with lipids, or what we commonly call fats. While proteins give us about 4 kilocalories of energy per gram, lipids give us a massive 9 kilocalories per gram. It's a real powerhouse. When digested, these lipids become fatty acids and alcohol, and they are absolutely essential. Without them, we couldn't produce molecules like phospholipids to build our cell membranes, or important hormones like estrogen and testosterone.
Moving on, let's talk about the unsung heroes of our biology: essential vitamins and water.
You see, we can neatly categorize our vitamins into two main groups. Vitamins A, D, E, and K are your fat-soluble vitamins. On the flip side, vitamins B and C dissolve right in water. Both types are incredibly vital for our metabolic processes. Take the B vitamin, riboflavin, for instance. It's strictly necessary for producing the very molecules that carry energy right inside our cells. Without it, the whole system just doesn't run.
And speaking of water, this is something you must always remember. Every single cell in your body is about 70% water by weight. It is definitely not just empty space. In fact, our blood plasma alone is 90% water. Even a tiny, tiny loss of hydration disturbs the functioning of our entire body. Hydration is the ultimate fundamental requirement for your cellular health.
Which brings us to our ultimate destination today, the basics of cell division. I want you to ask yourselves this crucial question. When you fall and scrape your knee, or when a plant drops a leaf, how exactly do those wounds heal? Or, simply, how does a living organism physically grow larger over time?
Well, the magic answer is cell division. It's an essential life process. It is how a brand new organism is formed, or how a complex multicellular organism grows and restores injured body parts. Now, this happens in two distinct ways: mitosis, which takes place in our somatic and stem cells, and meiosis, which is strictly reserved for our germ cells.
Let's explore mitosis first, the mechanism behind our growth and repair.
Before the division even starts, a cell prepares by doubling its chromosome number from 2n up to 4n. After that, the actual nuclear division, scientifically termed karyokinesis, begins. This beautiful process unfolds chronologically in 4 exact steps: prophase, metaphase, anaphase, and telophase.
If you observe closely, in prophase, the thin, thread-like chromosomes start to condense. They become thick and clearly visible, right alongside their sister chromatids. Then, moving into metaphase, the nuclear membrane just completely disappears. The chromosomes beautifully align themselves perfectly parallel to the equator of the cell, held tight by flexible protein threads we call spindle fibers.
Next comes anaphase, and this is where an incredible separation happens. The centromeres split. The sister chromatids separate and are actively pulled apart to opposite ends by those spindle fibers. It's quite fascinating because, under a microscope, these separated chromosomes being pulled look exactly like a bunch of bananas. Isn't that a fun way to remember it?
Finally, we reach telophase. The chromosomes arrive at the opposite poles and begin to decondense. A brand new nuclear membrane forms around each set. But we aren't quite done yet. After this complete nuclear division, the cytoplasm itself has to divide in a process called cytokinesis. A small notch forms right at the center, deepens, and voila, we are left with 2 distinct, perfectly identical daughter cells.
Now let's shift gears to our final topic, meiosis, creating new life.
Unlike mitosis, meiosis is a bit more complex and happens in 2 distinct stages. In meiosis 1, there is a truly remarkable event called crossing over, or recombination. This happens between homologous chromosomes. Basically, genetic material is physically swapped and mixed before the cells divide.
This mixing is absolutely crucial, students. It is the very reason why genetic diversity exists in nature.
When we look at the end result side by side, the fundamental difference becomes crystal clear. Mitosis gives us two identical diploid or 2N cells. Meiosis, however, pushes through a second stage to ultimately give us 4 genetically unique haploid or N daughter cells. Because of that crossing over we just talked about, every single one of these 4 cells is genetically different from each other and from the parent cell.
As we wrap up today's explainer, I want to leave you with something to really ponder. Think about the meal you just ate today. How are those exact proteins, lipids, vitamins, and water actively powering this invisible, incredible microscopic dance of chromosomes inside you right at this very second? Everything is intricately connected, from the macro food on our plates to the micro cells in our bodies. Keep observing, keep questioning, and keep learning.